differential neuroinflammatory gene expression in the brain from adult mice treated with neonatal sucrose. Worldwide, approximately 10% of infants are born premature, and many of these infants need to be admitted into the neonatal intensive care unit, or the NICU. And in the NICU, preterm infants receive a daily average of 7 to 17 clinically required but painful procedures. However, to mitigate this pain, oral sucrose is commonly administered as a non-pharmacological analgesic for minor procedural pain relief. But in mice, it's shown that neonatal exposure to repeated sucrose with or without pain has been associated with several adverse outcomes in adulthood. And this includes smaller volumes in white matter structures, lower levels of serum insulin like growth factor 1, poorer growth in females, and poorer short-term memory. In subsequent work, where neonatal mice were exposed to sucrose only, we profiled gene expression of 30 neuroinflammatory, metabolic, and neuromodulatory genes. And we found that sucrose-treated males tend to have lower gene expression relative to water-treated mice, while females show the opposite effect. So to further investigate these preliminary findings, we conducted targeted gene profiling of pro-inflammatory cytokines of brain tissue of those same adult mice exposed to neonatal sucrose or water. In the present study, we're examining the isolated long-term effect of sucrose exposure. Therefore, we've excluded pain from our experimental model. So the aim of our study was to investigate the levels of two pro-inflammatory cytokines, IL-1 beta and IL-6, in brain tissue of adult mice exposed to neonatal sucrose or water, which were the control groups. And we hypothesized that IL-1 beta and IL-6 levels would be altered in mice exposed to neonatal sucrose compared to the control groups. And these cytokine levels would be brain region specific and sex dependent. So for our methods, from postnatal day 1 to postnatal day 6, mice were provided with water or sucrose 10 times per day. And then from postnatal day 6 up until 16 weeks, where we did tissue collection, these mice were left undisturbed. At P21, mice were weaned onto a controlled diet and housed by sex in groups of 5 mice per cage with access ad libitum to water and food. Then to quantify gene expression of IL-1 beta and IL-6, we use qPCR in the left hemisphere brain tissue, specifically in the prefrontal cortex, hippocampus, cerebellum, and the striatum. We found a significant difference between male and female hippocampal IL-6 gene expression, suggesting the importance of sex in neuroinflammation. We also found a significant treatment by sex interaction in cerebellar IL-6 gene expression, where water-treated female mice had higher relative IL-6 expression levels than water-treated male mice. So contrary to what we predicted, neonatal sucrose exposure did not have a significant long-term effect on IL-1 beta and IL-6 gene expression in the adult mouse brain. However, this may be due to very limited sample size. Also, our sex-specific results add to our previous findings, suggesting that repeated neonatal sucrose treatment has long-term multi-level sex-dependent effects. And lastly, exposure to sucrose during the neonatal period may have short-term neuroinflammatory effects that require a subsequent challenge in adulthood in order to show these long-term consequences. And this has been seen in early life immune activation models. So for future directions, it would be important to investigate the effects of neonatal sucrose exposure on additional pro and anti-inflammatory cytokines, as well as neurotoxic factors such as TNF-alpha on the brain and serum gene expression levels in these mice. Additionally, it would be important to explore the effects of neonatal sucrose exposure in combination with pain on neonatal, juvenile, and adult pro and anti-inflammatory cytokine and insulin growth factors in the blood and the brain. Thank you so much.